Asexual reproduction in bacteria. Reproduction in bacteria is of two types, asexual reproduction and sexual reproduction. Asexual reproduction is a type of reproduction that doesn't involve the fusion of gametes or change in number of chromosomes. Right? Asexual reproduction includes binary fission, budding, and sporulation. Sexual reproduction involves a complex life cycle in which a gamete with single set of chromosomes combine with another gamete to produce a zygote that develops into an organism composed of cells with two sets of chromosomes. Sexual reproduction includes transformation, transduction, and conjugation. But today we will discuss only asexual reproduction in bacteria. So, binary fission in bacteria, it is a most common mode of asexual reproduction. Here, cytoplasm and nucleoid of bacterial cell divide equally into two following replication of DNA. Cell wall and cytoplasm split, resulting in formation of two daughter cells. Under favorable conditions, a bacterial cell divides by fission once in every 20 minutes. Right? So, various steps in binary fission. Step 1 is replication of DNA. Replication of DNA marks the initiation of binary fission process. The process of replicating nucleic acid begins from a replication origin. So, replication bubble is formed first. You can see here replication bubble. Replication bubble separates the DNA strands. Each strand then serves as a template for synthesis of the complementary strand. DNA or genetic material is then duplicated. You can see here, this blue colored DNA is a new DNA and black one, this is old, right? So this is replication, how replication starts. Replication bubble is an unbound open region of DNA where DNA replication occurs. Bubbles are created when enzyme helicase enzyme is present in nucleus, separates the two strands of DNA so that they can be replicated. Right? So here you can see a vegetative bacterial cell. One more thing, in bacterial cell, plasmid is present. So plasmid also copied, right? So each cell then receive one copy each. So plasmid, we know, is a small circular double-stranded DNA molecule that is distinct from cell's chromosomal DNA. As we know, plasmids naturally exist in bacterial cell. Plasmids provide bacteria with genetic advantage such as antibiotic resistance. When a bacteria divide, plasmids also divide and each daughter cell receive a copy of each plasmid. So, second step is growth of cell. After duplication of DNA, parent cell grows and increases its size. At the same time, two circular DNA strands migrate and attach themselves to plasma membrane in different sites. And you can see here, two DNA sets are present, right? And two plasmids are there. Third step, segregation of DNA. Following sufficient growth, as in second step, growth of the cell. After sufficient growth, cell elongates and starts pulling apart from opposite poles. A division septum created transversely in the cell, and the cell membrane extends and pinches inward, as you can see here. Invagination of wall occurs, and ultimately both invagination meets and a complete septum is formed, right? So during this process, separation of two chromosomes takes place. Fourth step is splitting of cells. Final binary fission step is splitting of parental cell into two daughter cells. Here you can see fourth step, two daughter cells are formed, each having a nuclear material of its own, right? So next is budding. Budding is a form of asexual reproduction in bacteria in which a new organism develops as an outgrowth or bud, because this is due to cell division at particular site. This is bud, and this is a bacterial cell, and bud, outgrowth, right? New organism in the form of bud, it is present here, remain attached as it grows and separating from the parent organism only when it is matured and leaving behind scar tissues. You can see buds may be present anywhere. Here the bud can also be here. So when it matures, separate from the parent cell and leaving behind scar tissues. These are scars. This is scar, bud scar, and in this case here, right? Since reproduction is asexual, newly created organism is a clone, means is genetically identical to the parent organism. Mother cell is retained here in this case, and new daughter cell is formed. This is mother cell, here mother cell, and new daughter cell is formed. Next process is sporulation, means formation of spores, right? In certain bacteria like Clostridium and Bacillus, cells tide over unfavorable conditions by forming spores, endospores. During this process, during sporulation, a portion of cytoplasm and a copy of bacterial chromosome undergoes dehydration. Dehydration means loss of water, and get surrounded by three-layered thick covering. Remaining part of the cytoplasm and cell wall then degenerate, resulting structure which is formed called endospore. This endospore can tolerate extreme environmental conditions and can remain viable for several years. When the environmental conditions are suitable, the endospore absorbs water. After absorbing water, it swells up and the wall splits, releasing the cell inside. It develops a new cell wall and starts functioning as a typical bacterial cell, right? So two types of bacterial spores are there. Endospores produced within the bacterial cell, and you can see here in this diagram. This is spore and this is cell. Example, Bacillus, Clostridium, etc. Exospores produce outside the cell. For example, Methylocyanus. So this is exospore. Stages of sporulation. Total 7 stages are there. Stage 0. Here you can see 0 stage means vegetative cell. Every condition in cell is normal, right? Stage 1 and 2. Stage 1 and 2 means asymmetric cell division. First, in this stage, bacterial chromosome become thread-like. You can see here, thread-like, known as axial filament, right? Axial filament attached to cytoplasmic membrane by mesosomes. Then asymmetric cell division occurs. Cell membrane forms septum, as you can see here, stage 1 and 2. The septum is formed near one end, which encloses a small portion of DNA, forming 4 spore, right? Next stage means stage 3rd, engulfment. Engulfment means, here you can see stage 3rd, engulfment, where mother cell membrane grows around the 4 spore, engulfing it. 4 spore now has 2 membrane layers at this 3rd stage. Stage 4, cortex formation. Here, chromosomes of mother cell disintegrate and exosporium synthesis occurs. 4 spore starts forming primordial cortex between 2 membranes. Stage 5th and 6th, here small acid-soluble spore proteins and dipicolinic acid produce incorporation of calcium ions with dipicolinic acid occur and forming calcium dipicolinate. Dehydration of cytoplasm also occurs at this stage. Coat layer is formed and maturation of endospore takes place, right? So last stage, release of endospore. Release of endospore. Cell lysis and release of endospore takes place here. So mature spore, and it again germinate and gives a new vegetative cell. So what is the structure of mature spore? Here you can see centered core surrounded by inner membrane, then germ cell wall. This is white part is cortex surrounded by outer membrane, and then thick coat is there, this black colored dark, and last is exosporium. So this is the structure of mature spore, right? Spore germination, next step. Endospore remains dormant for years, but under favorable condition, each endospore germinates to give rise a vegetative cell. So this process, spore germination, involves 3 processes: activation, germination, and outgrowth. Activation of endospore means the spore coat. Activation of endospore in this step, spore coat must be damaged by heating for several minutes, right? So this is first process, means activation of endospore. Next process is germination. Activated spore initiates germination after binding of effector molecules that activate autolysis and destroy peptidoglycans of cortex. Thereafter, water is taken up and calcium dipicolinic acid is released. Then outgrowth. After uptake of water, swelling of spore occurs and synthesis of DNA, RNA, and proteins also takes place during this process. A small germ cell emerges out after breaking the spore coat and begins to grow into a new vegetative cell, right? So this is all about asexual reproduction in bacteria.